Welcome back, class. This is week 1, part 3, fundamentals of phlebotomy, the circulatory system. We're going to be talking about the blood and its composition. All right, so blood is made up of, we've got the plasma portion, and then we've got what we call cells that are inside the blood. So we have the plasma, and this is made up of 90% water, gases, minerals, carbs, lipids, which is fats, proteins, waste products, vitamins, hormones, and drugs. Okay? And we would say that these things are dissolved in the plasma, meaning, it's kind of like if you add a powder to a liquid and it dissolves inside the liquid, basically. Okay? So these, go. Just remember that plasma is 90% water, and all of these gases, minerals, carbs, lipids, proteins, waste products, vitamin, hormones, and drugs, and they are dissolved. Okay? The formed elements. Okay? And we're going to talk about those. Those will be yours. All right, if we look here at this picture, you're going to see we have the red blood cells, we have a white blood cell, platelets. These are the formed elements. Okay, that means they have they're cells, right? They're not going to dissolve inside of the plasma. Okay, we're going to talk more about each of these cells as we go. All right. So the reason why I'm telling you formed elements, that's how you're going to see it on your test and on your certifications, okay? So you also are going to need to know the medical term for each of these formed elements, and we are going to go through that right now, all right? So erythrocyte, that is a red blood cell. And erythro, this part of the word here, refers to red, and cyte means cell. So, red blood cell. You also need to know that the red blood cells are the most numerous cells in the body, or in the blood. I should say blood here. And they carry oxygen and carbon dioxide, and you need to know that erythrocytes, or red blood cells, are produced in the bone marrow. Now, this is actually what they look like under the microscope. This is an electron microscope, so it gives a really awesome clear picture. But this is what your average lab tech's going to be looking at. And if they run a CBC, which is a complete blood count, you'll learn more about that as we go, and it comes up abnormal, they're going to check the results by manually counting under the microscope, and this is what they're looking at. This right here is a white blood cell, and we're going to talk about that next. All right. And then here in the middle, this is just a representation of how the oxygen is carried, okay? So we have hemoglobin, which are little iron molecules that attach to the red blood cell. Oxygen needs these hemoglobin molecules to attach to the red blood cell. Once it's been bonded, it's carried on where it needs to go and released to the tissues. Okay? All right. We then have leukocytes, or white blood cells. Okay? Leuko means white, and again, cyte means cells. These are formed in the bone marrow in lymphatic tissue. Their job is to neutralize or destroy pathogens. They can leave vessel walls and go attack the pathogens. There are 5 types of leukocytes. Now, this is an important statement. You will see it on tests and certifications. Leukocytes are the only blood cell or formed element that can leave the blood vessel. All right. So your red blood cells, they can only leave the vessel if there's an injury to the vessel. Okay? Your platelets can only leave the vessel if there's an injury to the vessel. White blood cells, their job is to attack and defend invaders of the body. And so they are capable of leaving the vessel walls to go attack the pathogen. Okay? And each of these pictures up here is a white blood cell here. Now, these are red blood cells, but these are white blood cells doing its job. This is a bacteria. These are bacteria. And see how the white blood cells like got little arms going out to catch it, and it will bring it back in and do what they call phagocytosis, and will eat and destroy the bacteria or toxin, whatever it is fighting within the body. Okay? Pretty cool what they can do. All right. Now, you don't have to know the 5 different types of leukocytes or white blood cells, but we are going to go over it. So you're just familiar, okay? So, like I said, there are 5 different types of white blood cells, and each of these are activated based on different pathogens that come into contact. They come into contact with. So, like your eosinophils, those are going to come into play with allergies. I believe basophils come into play with allergies as well. Monocytes are going to be like infections. Lymphos are going to be infections. You don't have to know these, just kind of be familiar with the terms. We have lymphocytes, neutrophils, basophils, and monocytes, okay? And these are what they look like in the microscope. So again, if they had a CBC done and they had an abnormally high white blood count, they're going to go manually count the white blood cells, and they're also going to look at the type. So that's going to give the technician the information he needs to give to the doctor as to what might be going on, okay? Thrombocyte is our platelets. Thrombocyte is the medical term for platelets, okay? Now, these are essential to coagulation. You need to know coagulation is the blood clotting process, okay? Without platelets, we could not stop our blood from clotting or stop an injury to the vessel from bleeding out, okay? This is what they look like under the microscope. I think they look like little glue dots. And this is what they look like under the really powerful microscope. So they're pretty cool. All right. So next, we know that everybody has a blood type, right? So um, you don't have to worry about this table here. So we know there's A, B, O. We have you have A positive, B positive, A negative, B negative, those types of things. Um, you don't have to know much about this other than the different types we have 4 different types of blood, and then we have Rh positive or Rh negative blood. We're not going to go into deep details about this. You have a lot of other information that you need to be learning. You do need to know that your blood type is inherited, and it is determined by the presence or absence of antigens on the surface of the red blood cell. Okay? And then a person's blood either contains or has the ability to develop antibodies directed at the opposite blood type. This is why it's so important when people get blood transfusions that they get the right blood type, okay? Because if the wrong blood type is given, then the when we give the blood to the patient, it can cause the blood to agglutinate, which is clumped together inside the body, or hemolyze, which when the it can cause the cells to like destroy each other basically, and these are transfusion reactions that can be fatal. So it's your job as a phlebotomist is to make sure you are absolutely following the procedures for drawing blood, for identifications and all the paperwork together, because we just do not want to give someone the wrong blood type. And we're going to talk more about drawing blood for these purposes when we get into special collections, okay? We also want to talk a little bit about hemolytic disease of the newborn. So this is when a mother is Rh negative. She is going to get a RhoGAM shot to avoid this. So if a mother is Rh negative, when she has her first baby, if the baby's blood is positive, it won't cause a reaction, but the next necessarily cause a reaction, but the next one would. And hemolytic disease of the newborn is where the mother and the baby's blood is incompatible, and it can be fatal for the newborn. And I believe it is dangerous for the mother as well. So when a mother is Rh negative, she will receive RhoGAM shots. Okay. Let's talk about compatibility test and cross matching. So, as a phlebotomist, you are going to need to be familiar with testing. We don't go deep into each test and the tube it goes in and all of that stuff because, quite frankly, 6 weeks is not enough time to learn all of that, okay? And when you get in your job, they're going to teach you this, and then you're going to get used to your facility and the same things that you're drawing over and over again, and you'll learn it that way as well. Right, but you do need to know that um, compatibility and cross matching is a test that determines whether the donor blood and recipient's blood are compatible or suitable to be mixed together. Okay, and the way that they do this, I used to work in blood bank in the lab, and the way that we do this is we will run the blood typing, find out what their blood type is, and then we take, if you look on these bags up here, there's these little pieces right here, and each of those have a corresponding number on that little tube that corresponds to the bag. And we'll take that, we'll run, and we know what the blood type is, it's listed on the label, but we're going to run that blood type again to make sure. We cut off these little thing, we call them pigtails, and we run the blood type on the test or on the blood again, and then so we have these two blood types. We are sure that according to our first test of blood typing, they are compatible. And then what we do, we look for their antibodies, and we want to make sure that the donor does not have antibodies to the recipient's antigens on their red blood cells, right? And the way that we do this, there's several ways, but one of the process before we're completely done is we actually in the tube mix the donor blood with the patient blood to make sure that it is compatible and it doesn't clump together and it doesn't hemolyze and destroy each other. And that's the last process before we say this is a compatible match, okay? Now, you all, as phlebotomists, will not be doing that actual testing, but you do need to understand the importance of it. All right. So our blood specimens, this is very important to remember. Okay, you are going to see this for the next 6 weeks. Very, very, very important. Okay, serum. That is the fluid portion of blood that remains after clotting. Okay, it can be separated from the clot by centrifugation. It does not contain fibrinogen because fibrinogen was used up in the clotting process. All right. Then we have plasma. This is the fluid portion of whole blood that has been separated from its formed elements, or red blood cells, white blood cells, and platelets, by centrifugation. It does contain fibrinogen because it did not clot. Okay? Now, this may seem a little foreign to you right now, but I'm going to explain a little bit. So we have all gone to the doctor and had our blood drawn probably at some point, and we saw all the different color tubes, right? Well, these tubes do different things to the blood. Some blood that we need to test, we need it to clot, and then we put it in the machine that spins it really fast, and the red blood cells go to the bottom, the formed elements go to the bottom, and then what is left at the top is the serum, okay? And then when we draw blood that is in a tube where the blood does not clot and we spin it in a centrifuge, the white blood cells, red blood cells, and platelets go to the bottom, and the plasma is what is left on top. Now, you need to know this. You cannot visually look at blood that has been separated by a centrifuge and tell the difference between serum and plasma. You cannot. That is a test question. You will see that again, okay? You cannot visually tell serum and plasma apart. You have to know if it was drawn in a tube that clotted or did not clot. Now, how do we know that? Well, we have a whole lesson coming up in the next couple of weeks and over the next couple of weeks where you're going to learn the tubes and which ones clot and which ones don't clot. Okay? All right. This is another breakdown of what we just talked about and is very, very, very important to remember. Blood that has not clotted, we say that it is anticoagulated. All right? And this is coagulated blood over here if it's clotted. Okay? Remember, we said coagulated means clot. So if it's anticoagulated, anti means against. So if it's against the clot, all right? I do want to talk about this right here. This little portion right here, when this is a whole blood specimen, okay? Whole blood. It did not clot. When we put it in the centrifuge, it spun it down. Here's the red blood cells, all right? And then right here, this layer, this is our white blood cells and our platelets. We called this the buffy coat, all right? It is important to remember that thin white layer between our plasma and red blood cells is called the buffy coat, all right? Very important to remember that. All right, here we go. So if you look at this, can you tell this apart? They both look similar in color, right? But they're not. This is a whole blood specimen. I know that because I know that the blue top contains an anticoagulant, which is a chemical that keeps the blood from clotting. Okay, an anticoagulant is a chemical added to the tube that keeps the blood from clotting. All right, and then this is the Buffy coat. Now, this is not correct. This is the Buffy coat, all right? I would say this person probably not doing too well. That's a pretty large Buffy coat. All right, then we have this side over here. This is serum. That's our serum, okay? This is our red blood cells. I know this is serum because this is a red top, and I know red tops clot. They are clotting tubes. They are coagulant tubes, okay? And these have additives in them that help the blood clot faster, okay? Now, when we draw the blood, we must wait at least 15 to 20 minutes before we spin the blood down. Spinning means put it in the centrifuge and separate it, okay? If you don't wait for the blood to completely clot, then you're going to get what is called a fibrin clot, and that is this right here. All right? This is like a ball of snot in the middle of our specimen. Now, the reason why that is significant is because that means the only amount of blood I can test is right here. Depending on the test, that is not a lot of blood. And you're probably going to have to draw the patient again, which is not fun for you or the patient, okay, or the doctors who's waiting on the results. So this is important to remember, that we always need to make sure that the tube has fully clotted, giving it at least 20 minutes, if not longer, a little bit longer. No more than an hour, though, because we don't want the cells to break down any of the components that we're trying to test. Okay, and we'll talk a little bit more about that in another lesson. So here, again, this is not going to be enough to run the test, especially if the test came out abnormal. The lab has to run the specimen again before reporting an abnormal result, because we want to make absolutely sure that we're given accurate results. Your job as the phlebotomist is to make sure that this specimen arrives to the lab in its best condition. And it's just so important to understand why we draw things in the tubes that we do, and then why we use the process and handling procedures that we do. Okay? All right. In order to get a plasma specimen, you must collect the specimen in an anticoagulant tube. And I just talked about that a little bit. All right. It's important to remember, okay? Whole blood. This is important to remember. Whole blood is in a tube, is the same form as it is in the bloodstream. So if we need to test to see what the blood is doing and what it's looking like as a whole, we would need a whole blood sample. For example, whole blood is needed for a complete blood count, because we are running that blood through the machine, and it's like passing through this camera as if there was a camera on the vein, counting all the cells as they went through. Okay? So, whole blood specimens, very important to know, very important for your job, for your certification, for your test, is blood that is not allowed to clot or separate. We do not want plasma in this instance. We want a whole blood sample. So it must be collected in an anticoagulant tube. Now, if we've collected this and it's sitting in the tube rack, the gravity takes over and those formed elements naturally fall to the bottom. So before we go to run the test or process it, we need to make sure that it is mixed a minimum of 2 minutes prior to testing. Now you're thinking, I'm a phlebotomist. I won't be doing the testing. There are some tests and some procedures that you will be doing depending on the office that you work at or facility you work at. And there's a lot of stuff that we can do that we're trained on, and it's really cool. All right. So this would be a whole blood sample. You see, this is not separated, right? That's exactly what it looked like coming out of his vein or out of the veins. All right. And here's another example of your Buffy coat right there. Okay. This little there. And then this is plasma. Okay. You may wonder why, we have so many slides on this topic, but it's very important, okay? Another fact that you need to know is blood is made up of 55% liquid, which is plasma. We'll say whole blood. And 45% formed elements. So it's a little more than half liquid, and the rest are those cells that we can see under the microscope, okay? This is important to know that when you separate blood, you get red blood cells on the bottom, Buffy coat on above the red blood cells, and plasma on top of the Buffy coat. That is when you separate a whole blood sample. I don't think I said whole blood just a second ago. All right? This is what it looks like separated. All right, that is it for the components of blood. We're going to move on to the next part, which is talking about the clotting process. I have separated these slides just so it can give you a little bit of a break and you're not getting so much information at once. So I will join you back for the next part.